You guys, can you guys hear me? Okay, great. So, my name is Lancer Hamilton, and I'm the tutor for pharmaceutical biotechnology. Give me 1 second. Let me share my screen. Okay, so as I said, my name is Lancer Hamilton, and I'm the tutor for pharmaceutical biotechnology. Now, I would appreciate it if you guys would introduce yourselves, so I would, like know exactly who I'm talking to. You can say your name and what you want to study in the future, or if you have any interest in pharmaceutical biotechnology, either in the chat or you can raise your hand. Now select someone to talk. Okay. Hello, Azaria. Hopefully I'm pronouncing that correctly. You want to study marine biology? That's cool. That does have a little bit of pharmaceutical biotechnology in it, depending on how you look at it. So you should find this lesson interesting. And Shadika, if you're here. Okay, so I'll just move on from now. Okay, so what exactly is pharmacology? So pharmacology studies the effects of drugs and how they exert their effects on the human body. Now, what exactly are drugs? So I'm sure you guys know of a few drugs, common drugs like DPH and Panadol. They're basically chemical substances used or intended to be used to modify um, a condition that your body is undergoing. So, for example, if you're feeling pain, Panadol is prescribed to you. If you're having an allergic reaction, then DPH may be prescribed to you depending on your age. So, yeah, drugs may be used for prevention, diagnosis, and treatment. So, in the example that I said with like DPH and Panadol, that would be for treatment, and prevention, of course, is to prevent the sickness from even happening in the first place. So, and finally, diagnosis. Okay, so there's two aspects of pharmacology that we look at. I know the terms are large and you might not fully understand all of them, but I'm going to give you like a little bit of an introduction to that. So the first one is pharmacokinetics. So this is basically just what your body does to the drug. So how it metabolizes it, as in how it breaks it down in the body, distributes it to ensure that the condition, is corrected or is lessened. And well, the second one is pharmacodynamics, which is what the drug does to the body itself. Okay, so pharmacogenomic is I don't want to say new branch of pharmaceutical biotechnology, but it's the more creative branch that we're getting into because of just how much opportunities lie within it. So it mainly composes the Human Genome Project and the molecular biotechnologies. So just combining those two aspects is what allows the branch of pharmacogenomics to be formed. So the Human Genome Project, it's just it's something that scientists aim to pursue from like the late 19th century. And when was it completed? I don't remember when it was completed, but they wanted to just fully understand the human genome. So all like what exactly each aspect of DNA does and how it affects different individuals, how it makes us different from each other. That's what they wanted to understand. Because if you have an idea of like how the human body works, how it's programmed, then you know how to treat it. Okay, so I mentioned DNA from the Human Genome Project. So I have an exercise that will give you some more information about DNA if you guys are not familiar with it. Are you guys familiar with it? Oh, Shadika, I'm just seeing your message. You like to study medicine? Okay, that's great. Okay, this is really good for you then. Um, this is a different branch of medicine. I merge in with more academic stuff. But yeah, you can pursue it. This is what I aim to pursue. Okay, so do you guys know anything about DNA, like any facts that you'd like to share? Anything at all that you know? Obviously, this is a structure. Okay, so I'm gonna allow you guys to take your time to respond, and I'm gonna start exercise. But still, let me know so I know how far you guys are along in your understanding of the lesson itself and outside of this lesson, like what you've been taught in school, so I can adjust it to suit you guys, adjust the lesson to suit you guys. Okay. You just know that it has the genetic information of the body. Okay, that's good. That's a good start. Okay, so remember that I said that understanding how DNA work is essential because you want to know what exactly the body does. And DNA prescribes like everything that we see in the body. It's what's necessary to formulate anything that exists in the body, from organs to cells, tissues, everything in your body is controlled and programmed by DNA. So understanding the structure, once again, is essential, which is the point of this exercise. So the overall structure is a double helix structure. So it's shaped like this. So it's like a spiral, but there's two, hence double helix. So it looks like a twisted ladder. Okay. So what is the name of the shape of DNA shown to the right, which we just discussed? It's not a trick question. Oh, and let me also share it with you guys in case you want to do it later. It should be in the classroom, but I'll send the direct link as well if you want to follow along from your screen. Okay, I have to go back. So we said that DNA is a double helix. So it's spiral in shape, and there's two spirals, hence double, and helix, because it's like a spiral. So this answer would be double helix. Okay, how many strands are in DNA?
Two.
Yeah, there. Okay, so each strand of DNA is made of repeating subunits called nucleotides. Can I open a Jamboard on this right now? Let's give you guys more of an example. Better one. Oh, or I can show you guys just a simple image. Okay, let's look for the least complex one. Okay, so ignore all the chemical stuff. Oh, this one is even better. So this is what they mean by a nucleotide, just for reference. So they're made up of repeating units of molecules like this. So nucleotides are the building blocks of DNA because they're made up, as I said, of repeating units of nucleotides. Okay, so here are all of the components needed to build nucleotides. So first we have the sugar. This is a phosphate group. And these are the different bases. Let's build another one. And when these combine, that's how we get the DNA molecule to form. Okay, I have to build 4 of them before we can move on. So the names are a little bit complex, but they're adenine, thymine, guanine, and cytosine. Okay, yeah, next. So what are the 3 parts of a nucleotide? Let me go back to before. Maybe that was a little bit too fast. So sugar, phosphate, and a base. Sugar, phosphate, and a base. When those come together, which you'll see later in the exercise, DNA is formed. So those were the parts of a nucleotide. We combined those 3 things, those 3 shapes, to illustrate how a nucleotide would look. So what were the names given to those shapes? That's what the question is asking, basically. Okay, let's look at this one. Oh, there's a sugar group, a phosphate group, and a base. That's what makes a nucleotide. So we're gonna select the answer that shows those 3 different groups: sugar, phosphate, and a nitrogen base. Let me know if this is a little bit too complex. I'm not sure where you guys are exactly. when it comes to biology. So if it's difficult to comprehend, just let me know in the chat. You can private message me or just in the group or speak up if you like to. Okay, so in the nucleotide shown below to the right, what is the arrow pointing at? Let's look at this image. So the circle represents a phosphate group. The pentagon represents a sugar, while the rectangle represents a base. Now looking at the question again, the arrow is pointing to the circle. The circle represents what? Phosphates, yes. Now we're pointing to the pentagon. Sugar. And the last one, the rectangle. Yeah, a nitrogen base. And there's 4 different nitrogen bases. So let me type that. Thymine, guanine. One second. Okay, my apologies. Okay, so those are the 4. Oh, and these topics that I'm covering, they're typically covered at the CAPE Unit 1 level. I mean, it's a little bit more complex at this level, but the general idea. So if you guys are not there yet, or you will be going into CAPE studies anytime soon, then this will provide a nice introduction. You guys are set ahead. So now let's put some nucleotides together to build a strand. So remember what we said before, that these nucleotides will combine to form the DNA molecule itself. Okay, so to the right, you can see one strand of DNA. DNA, however, is double-stranded, which is why we say DNA has a what type of structure? It was the first question that we did. What type of helix? A single, a double, a triple, quadruple? Yeah, a double helix. So only one strand is seen. So that's like one helix being formed. So we have to form the second one. And we call that the complementary strand. So they're complementary to each other because they can combine to form a completely different structure, a full DNA strand, DNA molecule rather. Okay, so the bases that we described, the bases that are here, oh, right here, that join to the sugar group, we already taught that there's like 4 different types. Adenine, which is the A, T for thymine, G for guanine, and C for cytosine. So these will specifically pair to each other. So A with T and G with C. So you can just remember it's at GC. That's the way that I always remember it. Okay. Or G and C, they look similar. So those go together. And A and T are left. So naturally those go together. Okay. So what did we say goes with T? And what goes with C? T. Oh, Why isn't it moving? Oh, sorry. I'm just reading that. They want you to show how the bond was formed. Hopefully you guys got that. So by adding the A with the T, we formed bonds between those bases. So A and T, there's like little dots right there, and we call those hydrogen bonds. I won't get too deep into that. I'm not sure how much you guys are familiar with chemistry. Okay, so we said G with C. Now note the difference. While A and T form two bonds, which we call hydrogen bonds, G and C form three hydrogen bonds. Okay, so naturally G goes with C again. Oh, okay. Sorry. Okay, so there's another bond that's formed between the two nucleotides that we just added to the structure. So it's between the circle and the pentagon above, and that just joins the nucleotides together in one strand, while the hydrogen bonds join like opposing nucleotides together. G goes with C, we form bonds again. Now, how many bonds are we expecting when C and G join together? Yep, three. Now, there's a covalent bond formed, and it's between the phosphate group and the sugar. A goes with T, as we said before. This time we're expecting two, and another bond is formed. G with C, and A with T. So now we have two strands. So to the right, let's call them by their base. So we have A, G, C, T, C, T on the left. On the right, we have T, C, G, A, G, A, opposite strands. So these are complementary strands of DNA. They're held together, like the strands itself, like the opposing strands held together by hydrogen bonds, as you can see right here, while the members of each strand, which is more right here, are held together by a different type of bond called a covalent bond. Okay, so what holds base pairs together as shown in the diagram? What type of bond? Hydrogen, yeah. Okay. What type of bond joins nucleotides to each other as shown in the diagram? So we're looking right here. I think I said this once, but it was on screen, so I'm not sure if you guys saw it or heard me say it. But it's a covalent bond. Okay, so which nucleotide will pair with the nucleotide shown below to the right? So you have to look at the bit. Oh, okay, Azaria, you are fast. Okay, so it's A to T, G to C. Okay, so which nucleotide will pair with the nucleotide shown below to the right? Yeah, it's G. Okay, what should we name ourselves? Let's give it a combination. Let's go. Okay. Should get like a certificate thing at the end. Oh, it's not letting load the certificate. Okay, it's not loading it this time. Anyway, yeah, so that's it for that exercise. I did want to share the certificate with you guys, but I'll figure out why it's doing that and send you guys individual ones later. Okay, big drawing. Now actually for both of you guys, you said marine biology and medicine. So this is necessary, of course, to you guys prospective careers. So this is a cell. Now this is the nucleus. Oh, okay. Okay, yes. This is the nucleus, which houses DNA, which is what we were just talking about. So yeah, everything that you see here in this single cell orchestrated by DNA molecules that we just formed. And these cells become a part of your body naturally. They are a part of your body. They form tissues and they form organs from your skin to your stomach, your throat, your nose. Everything about you is formulated by these cells, which are orchestrated and controlled by DNA. Just emphasizing the significance. Okay, so let's do a cell exercise. Oh, this is a complex one. Okay, so actually, all right, I'll just walk you guys through this one. It's something that you're gonna have to, you can't really look at the picture to fully understand. So I'll walk you through it. Okay, so we have endoplasmic reticulum. So looking at this one, let's find endoplasmic reticulum. So this is where it is. It's surrounding the nucleus. So let's look for one in the picture that describes that. Okay, let me exit slideshow from here. Okay, so it's a little bit easier. Okay, sorry. Okay, so endoplasmic reticulum surrounds the nucleus. So let's identify the nucleus first. So the nucleus in this picture, it's circular in structure and of course surrounded by some random scribbly things. So when you look at this picture, what do you think the nucleus is? We're looking for like a central circular structure. Oh, and the chat's gone. Hmm. Oh, okay. All right. The one in the center. Yeah, I understand what you're saying. Okay, so nucleus goes here. And it's moving. Cool. DNA is being made, doing its job. Oh, those circle things are pink circle things are lysosomes. No, actually. Wait, it depends on where you're talking. You could talk in right here, if you can see my cursor. Oh, you're talking about on the left. Oh, over here. Yeah. Oh, let me show you the image again so that Shadika can see what we're talking about. So yeah, these are lysosomes, just small circular structures. So you're right, that should be it. Where is it? So lysosomes. Okay. Oh, Slayer. Okay, good job, good job. And what about the endoplasmic reticulum? We're not sure. Okay, let's look at the picture again. So endoplasmic reticulum surrounds the nucleus. So the one with the pink dots, like the jiggly structure that we're seeing, that's the rough ER, while the one without it is the soft ER. So rough endoplasmic reticulum versus soft endoplasmic reticulum. And these pink things that are on the rough ones are called ribosomes. Well, they just turned blue. Okay, now the membrane is just what surrounds the cell. And okay, why isn't this? Okay, great. Cytoplasm is the liquid right here. Mitochondria, the powerhouse of the cell. Okay, so you guys seem to be familiar with the components of a cell. So are you familiar with some of like the functions of each component?
I can only remember some of them.
Oh, okay, then just go ahead and share what you remember in the chat, or you can speak if you want. I'll start off while you type. So the plasma membrane is what surrounds the cell, as I said. And it basically controls what enters and exits the cell. So it's like, think of like a security guard. So if you want, like, okay, think of like a gated community. And so all the members of the gated community, they do and go about their own business. While if you're an outside person, you're gonna have to go to the security and say and talk to them and be like, do you know anybody in there? You're gonna be like, oh, I know the nucleus, so can I go inside? And they're just like, oh, okay, we'll verify you, and they'll let you in. That's what the plasma membrane does. Okay, let me look what you said. Nucleus is the brain of a cell, yes, because it contains DNA, while the mitochondria is the powerhouse. So yeah, the mitochondria right here, powerhouse of the cell. Um, Lysosomes are digestive. Okay, see if I can state it more simply. They're like structures that are in charge of absorbing any waste product that the cell makes to keep cell content clean. The Golgi apparatus is known as like the packaging units. Oh, maybe I should type this. Packaging units. So if you want to send something to another cell, so let's say you want to go from gated community A to gated community B, then before whatever package you're sending can exit through the security gate, which would be the plasma membrane in this case, the Golgi apparatus has to package it to ensure that the security recognizes it and the security be like, oh yeah, that package is supposed to go to community B, which is another cell. Takes it, allows it to exit, and it goes to community B, which is another cell, and that cell's own security guard, another plasma membrane, looks at the package, verifies that this is where it should be, and lets it in. So think of like the Golgi apparatus as like a post office. Okay, and cytoplasm, that's just the gel-like substance that's inside the cell. It holds everything together. And that's it for the basics. Okay, so let's return to this one. Okay, so now we're going a little bit more into the general topic of the general topic of pharmaceutical biotechnology. Back to screen share. Okay. So there's three aspects: drug development, drug research, and clinical application. So this is essential because all the vaccines that we have been developed through pharmaceutical biotechnology, hence drug development, and drug research. Yeah, is also an important part. If you watch like those movies, like, oh, you know, Stranger Things, you guys probably know Stranger Things, where they have like clinical stuff and the kids are in like some facility and they're being tested and whatever. Totally unethical. Don't do that if you pursue this career. But that's what we mean by clinical application, like trying to understand like how it works on human beings and then applying it actually to treat. Drug research is also, well, I already described the importance of it, but it's especially significant now because of how much we know and how much we've come to believe that we can control. So there's like constant new research that comes out about cancer, about like Alzheimer's disease, dementia, or diabetes, hypertension. And these individuals, they're trying to fix or try to lessen the impact on those affected by the disease by either creating a cure or some type of treatment to extend their life expectancy or make their lives more comfortable, which is really cool. So, yeah, that's more of the parts that I'm interested in. Okay. So I spoke about how like drug research and like biotechnology itself has just transformed before. So we can see it especially when we look at the traditional method of treating patients versus the newer method. So before, when a patient gets sick, they would go to a doctor. The doctor asks for symptoms, and then a drug will be prescribed for those symptoms. So the scientists, the doctor themselves, they don't know exactly what is troubling you. They don't really know. They just know that you have a fever. Okay, maybe some Panadol. You have a headache, Panadol. You have swelling. Okay, maybe some DPH. They don't really know like what's going on, but they can treat the symptom to make you more comfortable and hope whatever is happening to you just stops. Whatever disease, affliction, whatever it is, stops by taking that medicine. Now, the patient either gets better or remains sick, and new symptoms can come about, or the same symptoms can come about, and they take more medicine. So, oftentimes, especially like earlier in human existence, you just have people that have these persistent symptoms just throughout the entire course of their life because scientists or doctors weren't able to identify what exactly was wrong. So they can't fully treat it, they can only treat the symptoms. And of course, that's dangerous because there can be something wrong with you without you having symptoms. So now this is our hope for the future, which I think it's like pretty applicable now, but it's like a mix. Well, our now stage is mostly a mix of the traditional with the futuristic version of medicine that we like to think of. Oh, someone else joined. Hi, Crystal. We're discussing pharmaceutical biotechnology. So we were just talking. Oh, okay. Crystal's gone. Okay, so hope for the future. Oh, and she's back. Okay, welcome again, Crystal. So yeah, hope for the future. So sorry for the sidetrack. That was such a rant. So the patient gets sick. The patient goes to the doctor, and the doctor checks for symptoms. When the doctor notes those symptoms, they're just like, I think it could be this. I could think it could be that, or it could be this. So maybe I'll just run some tests to figure out what exactly that is. And so they order all these tests. They may take blood, saliva, hair samples, whatever it is, to get an understanding of like what is happening to you internally, right? So then the doctor goes on after taking all these samples from you, and tests it and looking for different things inside your body that would let them know like what exactly is bothering you. So for example, if you have sickle cell anemia. No, okay, let me type that. Oh, it's okay. You don't have to be sorry. You don't have to be sorry, it's fine. Sickle cell anemia. Got it. So that's where like the cells, your red blood cells, they're shaped kind of weird. We call it a sickled shape. So usually blood cells are round, while sickle cell anemia makes them like a glass shard looking shape. Now that just affects so many different things in the body. So when a doctor takes blood and looks at that under a microscope, they can be able to tell, hey, you know why you feel so fatigued or why you're always out of breath? It's because your cells are sickled. You have sickle cell anemia. Now, when they know that you have like you have the specific disease, treatment becomes so much easier because now they know that, hey, for sickle cell anemia, we have this set plan. And if you take this set thing, then we guarantee that you'll be able to live a full and almost like unaffected life. So that's how significant this is. So instead of like the patient just like being treated for a numerous amount of diseases because you don't know exactly what is happening, which is what was in the traditional method right here. The new method that we're trying to fully instill in healthcare, it involves like actual testing. Like I can tell you what's wrong with you. And that provides more comfort for the patient because they know like, hey, this is what's wrong with me, and I don't have to constantly be wondering like what's wrong, if I'm gonna die. The doctor knows what it is. The doctor knows that I won't die. So you feel better and you live your life normally. And your symptoms are greatly reduced because it's specific treatment now. Okay. So remember we talked about pharmacokinetics and pharmacodynamics. So this is what we're getting into a little bit more. So pharmacokinetics is how your body deals with a drug. And that includes absorption. So, okay, I think this one is a little bit complex. Okay, let me simplify. So absorption is just basically where the drug is broken down and it's like administered to like around the body. So if I take Panadol, it enters my digestive system, stomach does its stuff, it gets sent to typically like the liver. The liver does some stuff, and it's distributed into the bloodstream. And so everywhere that I'm feeling pain now receives what was distributed from the drug, and I feel better. I don't feel a headache anymore. I don't feel any pain in my arm, anything like that. That's how it works. So from your digestive system to your liver works on it some more, distributes it into the bloodstream, and the distributed parts of the drug find whatever is affecting you. It just goes all over until it finds the issue, to describe it abstractly, and it acts on it, stops a headache, stops a fever, etc., etc. Now, the speed and ease of which absorption occurs greatly depends on number one, the person themselves. So you know that some people can be allergic to drugs, or some people their absorption may not be as well. Some people have like liver issues as well. So there's just so many different factors that make it hard for them to absorb drugs or even take drugs themselves, which is why genetics is also an important thing that we look at, because we have to understand how the human genome works, because it's weird that this random person can't take Panadol when like almost the entirety of the world can. So understanding it, shapes how we treat different individuals so we can help a wider variety of people. Um, the degree of absorption, once again, genetics, physiological conditions, like if you have liver disease, fatty liver, thing like that. It also depends at the site. So some drugs are taken orally, that's via your mouth, while other drugs are taken like topically, which is like over your skin, and it continues. Some are injected right into the bloodstream, etc. So obviously, ones that are injected right into the bloodstream, those are faster than all the others. While ones that are taken topically, it depends. And the stomach is typically the slowest. Okay. This is distribution. We talked about this. Okay, and we're at the end. So I do have an exercise. So I wanted to show you guys like how prescriptions are set out. So how doctors do it, and give you guys a chance to make your own prescription after like I tell you how to formulate it and stuff like that. Oh, wait, there was another exercise. There was another exercise. I'm so sorry. I think I forgot one. Okay, or was it a PowerPoint? Oh, it was a video. Yes.
Medication absorption is the movement of a drug from its site of administration into the blood.
I can't hear the audio.
Okay. Thank you for telling me. Let me see if I can fix it. Oh, and everything's gone. Okay. Okay.
They're
Do you guys hear this?
inactivating it,
Yeah, I can hear now.
putting it into bile for elimination from the body.
Start over.
The remaining amount of medication absorption is the movement of a drug from its site of administration into the blood. Oral drugs enter the stomach, where they either dissolve and pass through the cell membranes of epithelial cells lining the stomach, or travel undissolved through the stomach to the small intestine, which is the most common site of absorption. Here, drugs dissolve and pass through the intestinal wall. Oral drugs then travel through the portal venous system to the liver, where they undergo the first pass effect. During this process, the liver metabolizes some of the drug, either inactivating it or excreting it into bile for elimination from the body. The remaining amount of active drug leaves the liver and reaches general circulation and target organs. If a drug is administered via intravenous injection, it passes directly into the bloodstream, thus bypassing absorption in the GI tract. If administered through intramuscular or subcutaneous injection, the drug enters either muscle or subcutaneous tissue, where it passes through gaps between cells into capillary walls, and then into general circulation or target organs, also bypassing absorption in the GI tract. Bioavailability is the net amount of a dose of a drug that is actually absorbed into the bloodstream. The bioavailability of oral drugs is less than 100% because of the first pass effect of the liver.
Okay, so I just want you guys to have a basic understanding of it. Okay, so yes, do you guys have any questions about anything we learned? Anything that you're interested in? Okay. Oh wait, I see messages. No. Okay. Oh wait, there's one more. Okay, no more. Okay, so I'm gonna stop sharing for now, and you guys are gonna hear me elaborate a little while I look for the pharmacology thing. Okay, so there's this new type of drugs that scientists have been really interested in, which is tailor-made medicine. So you know, like how you can just go to a store and be like, hey, give me that DPH, hey, give me that Panadol. Or like with a doctor notes, you can get what you want, but it's like on the shelf. So anybody that is prescribed it can get it. However, tailor-made medicine, it's more like I understand your genetics specifically. And so now that I know you specifically, I will treat your body specifically. I know how much your body absorbs medicine at any given time. I know how your body processes it, if it's different. I know everything about you that could affect how this drug works and how whatever disease affliction is affecting you works on your body itself. Now, once I know that specifically, because diseases vary in persons and they can mutate in persons. That's why we have so many different strands of like corona, the coronavirus, COVID-19 itself, influenza, so many different strands because of how differently it affects different people and how we can mutate based off those differences. So if I can understand you as a person specifically, just like how your body works and responds to drugs and how your body is reacting to the disease, then I can treat you more specifically. So like the risk of like um inaccurate treatment, like your body doesn't respond to it, or there's an allergic reaction, pursuing tailor-made medicine completely like disrupts that. It just throws it out the window. You know that it's going to be 100% effective because it's tailored to you. Well, that's really cool. Glad I'm alive to see it. Medicine specific to you will make your life easier. Is it gonna be millions of dollars? Oh, for sure. But yeah, still interesting. Why can't I find it? Okay. Give me 1 second. I might have to. And the twisted, based off her symptoms, the low blood count and the twisted ankle, you've noted that she has a ligament sprain and anemia, swollen ankle as well. That is affecting her ability to walk. Let's go over that one more time. So her name is Michelle Williams. She's 24, a female, pregnant in her second trimester. You are the OBGYN. You work at Farm to Clinic. She met with you today, and it's a routine visit. she has a low blood count, and she told you that she twisted her ankle prior. He looked at it, and he tested it, and he realized she has a swollen ankle overall, making it difficult for her to walk. And it happens to be a ligament sprain specifically. And for the low blood count, you've reduced it as anemia. Okay. You should have some idea of what you would prescribe, but I'm going to give you some information to help you out. So you prescribed her for the anemia. Oh, that message. Let's see if I was going too fast. Okay, and then, okay. So you prescribed to her iron sulfate. I'll write that if you guys don't know it. I need to prescribe 100 milligrams that she's supposed to take 3 times a day for 3 months. So the first prescription, like the first drug that you prescribe, iron sulfate, 100 milligrams, 3 times a day for 3 months. Now you prescribe Panadol, 1 gram, 3 times a day for 2 weeks. That's Panadol, 1 gram, 3 times a day for 2 weeks. And that one obviously treats the sprain. Okay. So I'm gonna give you guys some time before I share like what it should look like. If you guys would like to do it together, that's fine as well. Just message me. Oh, another person joined. For Malik, we actually just went through a number of things actually, but specifically how to set up a prescription. So that's the exercise that we're doing now. Oh, my bad. Too excessive. You wouldn't get that part. So I didn't go through that. But we did talk about 1 and 2, which is the iron sulfate and the Panadol. Now, for the Q8HR, super complex. But if you had like tri-di, as in 3 times a day, or just 3x, that's also correct. Same for number 2. If you just had 3x or tri-di, because omni is 1. That's 2. Yeah, tri-di should be for 3 times a day. Yeah. And that's okay. It's your first time doing it. And yeah, that's it. The rest of the information mostly made up, like the address, the contact, the patient number, all that stuff made up. That's it. And your signature, and remember your line. Okay, that's it. Next class we have the biotechnology activity, like drug designing activity rather. And we're going to do like extraction of DNA. So I shared those PowerPoints in the classroom of what exactly you would need. So that's for you guys to look at. We can talk about like the cancer research another time. Okay, that's it. And that's the end of my lesson. Thank you guys being here. So bye-bye. Have a good day.